Glenn asked me to try and make DNA barcoding sexy for you. So this is my attempt, and I apologise ahead of time. So I'm going to talk about difficult fungi, which is pretty much all of them. And what we've been doing as a group to sort of enhance our recording capability through using DNA barcoding. Oh, that didn't work. Oh, that did. So I'm going to just mention the Grampian fungus group, which Glenn's already mentioned. Say a little bit about what a difficult fungus is. Talk about some of our classic tools available to us for identifying fungi, such as the keys and the challenges around using them. And then I'm going to do a very rapid run through what kind of workflow we use for DNA barcoding. And then ultimately I'm going to mention a project that we've been focused on for the last few years around brittle gills, to give you an idea of how we're using that DNA barcoding. Grampian fungus group, as Glenn says, a very welcoming group with quite a large membership group now, and there's a core group of us go out and foray every autumn, and we do spring forays and winter forays to record fungi across the region. We've been around for more than 25 years, and we're mostly amateur field mycologists, although we're very privileged in having a couple of professional mycologists associated with the group. We're also British Mycological Society, BMS affiliated, which gives us certain advantages. I won't go into that, but that society affiliation is quite important. Our aims, as you can imagine, we like recording fungi, we like to encourage an interest in fungi, and we like folk to come out with us on forays. We give talks, we do workshops, all aimed at trying to enhance the way we're recording fungi across the region. We are also really keen to promote conservation of fungi and also, importantly, habitats of fungi, and I'll say a little bit more about that later on. To become a member, it's cheap as chips, £10 a year, and for that you get access to forays and a newsletter. And we also run a Facebook page and share some of the things that we're finding on that. The one thing we don't do, we don't forage, and that's becoming increasingly important in terms of us getting permissions to go into areas to record. So what's a difficult fungus, I hear you ask? Well, pretty much all of them is the answer. So I'll say a little bit more, but really I guess it doesn't really matter what you're recording. We've just heard about bumblebee identification, how challenging that can be. With fungi, just about every family and every genus has difficult groups of species that are really challenging for normal day-to-day identification. But I'm going to give you the chance to have a go. So have a look at these 6 photographs, and once you've digested it, shout out how many species do you think there are. I'll give you a few seconds. Anyone like to hazard a guess?
1.
1, 2, how many? 5. Any advance? 3, 8, yeah. This is pretty much how we feel as a group. So there are 2 species in these 6 photographs. The photograph in the middle on the left is one species, the one with the pale brown cap. Everything else is a single species. And it really kind of gives you a sense of how challenging it can be. You can have different species that look very, very, very alike, but you can have huge variability in other species. And I don't know if that's unique. Well, it won't be unique, but it's a real challenge for us in the fungal world that you can have such variability in how things look. We do, however, have really good resources. There's lots of keys available to us. And we also use not just what fungi look like, but we have microscopy to fall back on. And it's essential when we're using some of these keys to have microscopy available. But one of the challenges that we face as a group is some of these keys were written 20 years ago, 40 years ago. And what's happening, and this is because of DNA barcoding, is there's a really rapid change now in our understanding of species. And that's coming through research groups using DNA barcoding to basically look at relationships within different genera. And what we're seeing is classic splitting and lumping going on. Things we used to think of as one species are now 4 species. Things we used to think of as 4 species are now 1 species. And this is a really rapidly changing kind of thing for us. So we have to be aware that those changes are taking place, and it means we also have to dip into research literature to kind of come up to speed with what's going on in any particular group. So it makes it a challenge for us. But of course, it's really, really good, and it's really valuable stuff going on. So that brings us to DNA barcoding as a tool, and a tool that's becoming much, much more accessible for citizen scientists. So what we do, barcoding really just means we're looking at a tiny bit of the genome. And in fungi, that little bit is called the ITS region. It's about 600 to 700 base pairs long. Base pair is within the DNA helix. There are pairs of nucleotides, as they're called, that are joined together, that keeps the DNA structure, and that's what we mean by base pair. And DNA barcoding is all about getting hold of that little bit of the genome, and it is really a tiny bit of the genome, making lots of copies of it, which we call amplification, and then sequencing that, and then ultimately comparing that with reference data. So I'm going to take you through the steps that we use to do that. But first of all, I'll introduce you to Benny, the Bento Lab. I know, really creative naming there. The Bento Lab is kind of revolutionising this for citizen scientists. It's about the size of a briefcase, really light, comprises 3 main components. In the middle, you've got a centrifuge for separating out organics. On the right, and I'll show you some more pictures, is what's called the thermocycler, and it's a heat block essentially. On the left, you can see the orange gel electrophoresis tank, and under that is a lamp, a UV lamp. So that's basically it. What Benny allows us to do is extract and amplify DNA, but it doesn't allow us to sequence the DNA. We have to have that done by a professional lab. So I'm going to quite rapidly just take you through the kind of workflow that we operate when using Benny. First of all, we have to get hold of our DNA, which you might think is really easy, but can be challenging. We take a tiny bit of flesh from a fungus, we grind it up, and then we take an even smaller part of that. You can see the yellow ring is showing you just how much material we actually need. It's tiny. We put that in a lysis buffer. We heat that up in the thermocycler. And what that does is just basically pop open all the cells and releases the DNA into solution. We then neutralize that, and that's called a DNA template. That's just DNA floating around in solution, and we can freeze that. We can keep it frozen for up to 3 years, so we can use it any time we want to, which is really helpful. And then the tricky part of the whole process, which is actually amplifying that little bit of DNA that we're interested in. And this is where the thermocycler comes into its own. It's a programmable heat block, so we can set it up how we want it and let it run. We take our little bit of DNA template and we add it to a mix called a PCR mix. And that PCR mix contains a primer. And all a primer is a tiny little bit of DNA. And it acts a bit like, I kind of think of it as a key to unlocking the whole process. It attaches to our DNA template and it makes copies just of the little bit of DNA we're interested in, the ITS region. What we do, we put that mix into the thermocycler, it heats up, the DNA unzips, the primer attaches, it copies the ITS region, then we heat it again, we do it again, and we keep doing it for 30, 35 cycles, and that amplifies that amount of ITS DNA in solution. We end up with hundreds of thousands of pieces of ITS DNA. It takes a couple of hours to do that, and we finish off, if you look bottom right, you can see that's a little collection of PCR product waiting to be sequenced. Now you're thinking, because we all do, there's got to be loads of DNA in there. But there might not be, because PCR can fail for lots of different reasons. So we have to check we've actually got DNA. And we do that using the gel electrophoresis tank. It's a little plastic tank. It's got a negative and a positive electrode. We fill it with an agar gel. We then pipette small amounts of our PCR product into it, plug it into Benny, leave it for an hour, and then we use the lamp on the lab to have a look to see whether we've got any DNA. The photograph bottom right shows you what kind of thing we would hope to see. And you can see some really bright blobs across the middle. That's our DNA. You can see some fuzzy blobs along the bottom. That's just residue from the PCR mix. And then along the side, you can see some fine bars. And that's a pre-supplied thing called a DNA template. And what that's doing is giving you an idea of how long the strands of DNA are. As the DNA passes through the gel, it gets stopped as it goes, and that stop point relates to how long that bit of DNA is. So the nice thing about this gel plate is that you can see there's some really bright blobs there, and that's telling us we've got lots of ITS. Well, it's telling us we've got lots of DNA. And then if we compare it to the DNA ladder, which we can calibrate, it tells us how long those pieces of DNA are. And in this example, it's between 600 and 700 base pairs. So hooray, we've probably got ITS DNA. You can see there are some slightly fainter lines there. There's still ITS DNA there, so we can still send that off for sequencing. But you might notice there are some gaps in there. There's two columns that have no DNA showing, and for whatever reason, that PCR process has failed for those two samples. So we can't do anything with them, chuck them away. We're not going to get a sequence from them. The ones where we can, where we've got DNA, we then send that off for sequencing. And the British Mycological Society has a relationship with the University of Aberystwyth. We send them off to Aberystwyth. A couple of days later, we get our sequences. This is the kind of data we get. Yeah, who knows what it means? We get a raw data file and we plug it through a piece of software, a free bit of software, and it produces this chromatogram. And this is what a sequence essentially is visualized as. And it tells us lots about the data. The most important thing is it's telling us the quality of the data. So you can get a sequence, but it can be rubbish quality, and there's very little point in doing anything with it. Or you can get really high quality sequences, and this is a pretty high quality sequence. We do get junk data at the beginning and end of a sequence, so we just chop that off, we just edit it, doesn't matter, throw it away. We don't want that confusing the results. And we end up with just this string of letters. After all that, that's all we get. But that's the goal, that's the solid goal, that's what we're after. That is a sequence, and it's an ITS sequence of one of our samples. What do we do next? We need to compare that sequence with reference sequences, and this is a really important step. We've got several resources. Some of them you'll be familiar with. There's a thing called GenBank, which is managed by the NIH in America. Thousands and thousands and thousands of sequences from all manner of things are in that database. The other one is Unite, and this is focused on fungal ITS sequences. So what we can do with our sequence, we just copy it, We paste it into the query box, we hit submit. Within 20 seconds for GenBank, or within a couple of minutes with Unite, we get an output. And this is what it looks like. And it's giving us a list ranked by how similar our sequence is to the reference sequences. And you can see in this one, I've put a blue circle on here. This is pretty good, this one. I mean, it's pretty sure that this is what we're actually collecting. The percent, anything above about 99% is really similar. Anything less than 99%, we probably need to question and have a little look at and more careful examination of. In this case, this sample pretty clearly one particular species, and we go back to our field data and our microscopy and what we've written down in terms of ecology, just to sense check this. So it's an integrative approach. We're not solely relying on the DNA. And we would then have pretty, we'd be pretty certain this is what we've got. So we would then record it as this. Sometimes these results are a bit more of a mixture. That can be for lots of reasons. It can be that the taxonomy hasn't been worked out, so people are naming things differently. It can be because there's no reference sequence in the database, and that doesn't mean that species hasn't been described yet. But in rare cases, it could actually be that you found something that's new to science, and it simply isn't there because no one's found it before, which is quite an exciting thing, but quite rare. So this gives you a really good way into trying to figure out what you've got. And we've been doing a project jointly with Andy Taylor. I don't know if Andy's here today, I've not seen him, at JHI over the last 5 years. We've been focused on a particular genus, the Russula genus, or brittle gills, which I'm sure you're all familiar with. You will have seen them as you've been out and about in the autumn in our woodlands. We've collected and processed more than 200 specimens, and we now have nearly 170 really high quality ITS sequences. And we've confirmed the presence of 61 species just in this genus in our region. That's about a third of the British total for this genus, and it's pretty good, I think. There are more to find, I'm sure, but we're pretty much there, I think. And we've discovered many nationally rare species in our region, and also a likely new species for Britain, so not previously been recorded in Britain. We're also building up a better picture for this one genus of what are our best sites in the region, and I think that's really important because a lot of those really high diversity fungal areas are not protected in any way. And management can do quite significant damage to the woodland floor and what's growing there, including the fungi present. So we're quite keen to share that knowledge and help people understand the value of some of those sites for fungi. It has done what we set out to do. It's helped us improve our identification approach, given us a new tool, a really good tool to help us do that and to improve our recording. But there are still some difficult fungi out there. So not only are we continuing to look at brittle gills, but we're expanding our range and we're starting to look at things like webcaps, coral fungi, and a whole range of other fungi. We've got a bit of a focus on Caledonian pinewoods and birchwoods because they're nationally, our region is really important for those. So we're going to keep working through and using Benny to help us enhance our recording. And I'd just very quickly like to thank a few folk, Andy, who I've mentioned, Glenn for his support, in particular helping us to secure the Bento Lab with support from Aberdeenshire Council through the mechanism that Glenn mentioned. A few folk you won't know, and also the British Mycological Society, but also GFG members, and thank you for listening.